for your interest in hearing more about the Aptose story. My name is Avanish Vellanki, Chief Business Officer of the company. In the interest of time, I'll jump right into it. As a publicly traded company, we will mention that we are going to be making forward-looking statements throughout the course of this presentation. So who are we and why are we interesting? It's impossible to tell the Aptose story without mentioning that Aptose was born out of another biotechnology company called Loris Therapeutics out of Toronto, Canada. Now, Loris Therapeutics was developing this program, LOR, at that time known as LOR-253, for patients with solid tumor cancers. However, when the new management team took over, we had a very different approach based upon the mechanistic rationale of 253 for patients with blood cancers. Apto-253, as it's now known, is an inducer of the gene KLF4, or Kupffer-like factor 4. And new insights into the biology of AML has shown that suppression of KLF4 serves as a key leukemogenic trigger in patients with AML. And as luck would have it, Loris, the small company in Canada, had the only inducer of this gene that we were aware of in the industry. So we had felt that we had an opportunity for a first-in-class therapy with the opportunity to leverage novel biomarkers to create a truly personalized therapy for patients with AML. And that fundamentally is why we were so excited. We took this message to the street and ultimately built a new management team around the story. We built an entirely new clinical development team. We now have offices in San Francisco, Toronto, and in San Diego. We were able to share this message with global investors in biotech, raising $45 million across the United States, and ultimately changed the brand to Aptose Biosciences to rebrand ourselves from the prior Loris legacy. We ultimately listed on NASDAQ under the ticker APTO and uplisted back in October of last year. Here is the new management team led by Dr. William Rice, Chief Executive Officer and Chairman. We also built a marquee list of clinical development experts, including the likes of Dr. Daniel von Hoff, who is known for his role in commercializing various breakthrough oncology therapies. Dr. Brian Drucker, known for his role in the development of Gleevec for Novartis, along with Dr. Michael Andrea from MD Anderson Cancer Center, and Dr. Stephen Howell, who is a distinguished professor from the Moores Cancer Center at the University of California, San Diego. Now, we only have time today to talk about the lead program, Apto-253, but we do have other programs in the pipeline. And as I mentioned, while the program was initially developed for solid tumors, we have moved it into blood cancer patients based upon this new mechanistic rationale and the opportunity to leverage these biomarkers for targeted therapy. Now, many of you already have heard a lot about AML as an indication, and you know that it is an indication that predominantly affects the elderly with an abysmal 5-year survival rate. Now, many of you also know that the standard of care in this indication are conventional cytotoxics, cytarabine and daunorubicin, that have significant toxicity and adverse events. And therefore, it comes as no surprise that the industry has moved to looking for next-generation therapies that are more tolerable, targeted regimens. However, this has been plagued by what has been deemed as an indication that has extreme heterogeneity and no underlying unifying mechanism that drives AML as an indication. So this was until a couple of academic journals, a couple of academic papers were released, first in 2007 and then in 2013, that showed that there was a potential underlying cause to AML. And this is the second of the two papers that showed that CDX2, an embryonic gene, could be playing an operative role. CDX2 is involved in the constitution of the hematopoietic system, but is effectively silenced and turned off prior to birth. And it has no effective expression in any normal adult. However, in the vast majority of patients with acute myeloid leukemia, this gene is effectively turned on across all subtypes of AML. And these papers also noted that CDX2 ultimately led to the downregulation or the silencing of KLF4. So we put together a very modest cartoon that depicts what we think is happening. A normal hematopoietic cell exhibits the signature of no CDX2 expression and KLF4 transcription. But over time, various things happen as a result of aging, various alterations, potential mutations that lead to the flipping on of CDX2 as a gene. It's not known entirely why this happens. CDX2 protein then looks for and binds a specific region of the KLF4 promoter. Upon binding, CDX2 recruits in KDM5B demethylase, and this leads to demethylation of histone 3 lysine 4. And that demethylation is what is assumed to lead to the suppression of KLF4 and the AML phenotype, with CDX2 on and KLF4 effectively off. These two papers also went further to look at this AML phenotype, CDX2 on and KLF4 off. And what they did in the course of this research was to transduce in constitutively active KLF4. And what they saw was a variety of downstream effects. They saw the induction of P21, cell cycle arrest at G1, induction of caspases, and the annexin 5 staining, indicative of programmed cell death. The theory was, with this agent, APTO-253, we could pharmacologically lead to the same result, all of the same downstream activity and cell death. This was concept when we all joined the company, and it was our objective to show whether or not this was going to result. So what we did is we actually sought out an AML-specific cell line, and we treated it with APTO-253, and what we saw was very interesting. We saw a dramatic increase at 16 hours of KLF4 levels. We saw an increase of P21. We saw cell cycle arrest at G1, the caspase induction, and the staining indicative of programmed cell death. So 253 effectively recapitulated genetic transduction of KLF4. We went further and looked at the activity of 253 against a wide variety of cell lines, both solid tumor as well as a variety of heme cell lines. And what we found was, indeed, the 253 was far more potent in AML cell lines than in the solid tumor lines. The IC50s were anywhere between 7 and 300 nanomolar, up to 1,000 times more potent than in the solid tumor indications. Now, what was interesting is, again, this company had previously completed a phase 1 study in patients with all-comers solid tumors. And what was identified from that study was that they were able to safely achieve exposure levels of between 2 and 6 micromolars, which is in this vicinity here, exceeding that which we think will be effective in leading to AML cell death. Now, a little more information about APTO-253. It is a small molecule. We have IP coverage through 2028 for composition of matter and use. We have seen significant single-agent activity across a variety of AML cell lines as well as AML subcutaneous tumor models. And what is perhaps equally as exciting is that we have also shown activity, not just additivity, but synergy with a variety of additional agents that are part of the current standard of care, cytarabine, daunorubicin, as well as a variety of hypomethylating agents that are currently being used as off-label therapies for patients with AML. Now, what's also very compelling about this program is that it does not affect bone marrow, and therefore the non-myelosuppressive nature of this therapy, we believe, could lead to very robust activity, not only as a single agent, but in combination with a variety of other targeted and other cytotoxic cancer therapeutics. We have filed IP for the use of 253 in conjunction with the CDX2 and the KLF4 biomarkers, as well as P21. And we hope to employ usage of this for the selection of the optimal patient population in the future clinical developmental plans of APTO-253. A quick word about our clinical developmental path. We have already begun enrolling patients in a phase 1B dose escalation study across 2 arms, AML and high-risk MDS, as well as lymphomas and multiple myeloma. Based upon the outcome of this phase 1B dose escalation and upon achieving the recommended phase 2 dose, we expect to look back at markers of efficacy and then stratify these patients based upon their biomarker status. This will be a single agent expansion cohort, which we think will be across AML and the MDS patient populations. We also, given the non-myelosuppressive qualities of the drug, expect to pursue a pretty rigorous combination strategy for the agent. We are enrolling across 4 centers today with numerous additional sites planned. All of these sites are harvesting bone marrow and peripheral blood to identify those optimal patients based upon biomarkers. We have also are participating in what is known as the Beat AML initiative with the Oregon Health and Sciences University to look at hundreds of primary patient cells and the activity of 253 as a single agent, as well as in combination with a variety of other agents. And we hope to see data from that over the next several months. The phase 1B dose escalation study is dosing twice a week, every week on days 1 and 2. We've already dosed patients up to 66 mg per meter squared. And the next dose level is 100 mg per meter squared, which we believe is entering the therapeutic range for these blood cancer patients. At the 100 mg per meter squared, we believe we'll be getting into the low micromolar levels of exposure. So in summary, Aptose has been through a tremendous transformation over the last 2 years. We have built an experienced executive team, a clinical development team. We have taken the 253 program out of the solid tumor setting and targeted towards patients with AML and other hematologic malignancies. And we believe this could truly become a targeted therapy with a companion diagnostic. We have successfully raised over $45 million of new capital from blue chip, long-term fundamental biotech investors. We've listed on NASDAQ. And looking forward to near-term news flow, we have already announced publicly that we expect to provide an update on the status of the phase 1B dose escalation study around the time of the American Society of Hematology meeting in December of this year. We have also announced that based upon the tremendous interest of Aptose 253 and the KLF4 induction strategy, we've been approached by numerous other companies, clinical stage companies, for drug combination studies. And we expect to have that data over the next several months. And finally, from a BD perspective, we hope to build a comprehensive oncology pipeline with a stage list of assets targeting multiple cancer therapeutics.